in 1962, almost 9 years after the model was published and fiber diffraction images were published, uh, Crick, Watson, and Maurice Wilkins got a Nobel Prize for Physiology of Medicine in 1962. Now, let's just hear Watson. He is fairly senior at the point this particular interview was taken. Let's just hear his voice, because he is one of the very major figures of biology in the last century.
I guess my chief advantage came from coming from the field of genetics, where the big question is, what is the gene? And I sort of boxed myself in, and that was the only problem worth solving. Whereas Rosalind was trained as a physical chemist, Morris as a physicist. They didn't see themselves, as having only one objective. So my advantage, one was, it was the only thing I wanted to do. And second, the other advantage was I had a collaborator who was Francis. So one can't underestimate the importance. If I'd come to Cambridge and Francis wasn't there, I wouldn't have found the structure of DNA. I wouldn't have, had someone to talk about the meaning of the X-ray diffraction. And Proust and Kendall were interested in proteins. So Francis saw DNA as tremendously, or I should say, Francis saw DNA as potentially, the most exciting molecule around.
All right, so that was James Watson sitting here in Cold Spring Harbor Laboratory. He joined Cold Spring Harbor Laboratory and was his director for a very long time, and he settled over there. Major role in setting up leadership in that particular institute. Francis Crick went to Salk Institute in San Diego, and there he set up again a very, very nice set of units. The actual structure of DNA came fairly late. There's a paper in 1979 about the Z form of DNA, which I'll talk about, and another paper in 1980 from Dickerson's lab, where the first turn of bDNA was solved. Now, these structures came almost 20 years after protein structures came, and part of the reason was that DNA is a very flexible and a very floppy molecule, and it's this floppiness of DNA which makes it extremely difficult to crystallize, which is why fiber diffraction was the means to gather data on DNA in the. So let's now go into the chemical structure of DNA. Here is the DNA double helix in front of you, and in blue, orange, green, and red are the bases, which are inside the double helix, with the phosphate backbone outside the double helix. This is the way the backbone looks: phosphate, sugar, phosphate, sugar, phosphate, sugar, with the bases sticking out. And these bases sticking out form hydrogen bonds with an opposite base to make the DNA double helix. And effectively, what is holding these two strands together are hydrogen bonds. You can see these hydrogen bonds as dotted lines between adenine and thymine, which is a double bond, and between guanine and cytosine, which is pretty much a triple bond. So the double helical structure of DNA consists of two antiparallel sheets, and these two antiparallel sheets are antiparallel because one is running to a 5' to a 3' orientation, and the other is running in completely the opposite orientation. Right? Because some of you asked, here is a little bit more detail. These are the 4 different nucleotides, with the base, the sugar, and the phosphate basically circled over here. This is deoxyadenosine triphosphate, which is polymerized along with the other nucleotides to make up DNA. This is DGTP, I'm using the small form, DCTP, and DTTP. Okay, this is the sugar itself. And so one of you asked me a question about labeling. So this is the labeling of the carbon atoms in the sugar: carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5. And the polymerization of the nucleotides is happening from 5 prime to 3 prime direction, which is basically this direction, which is why we say 5 prime to 3 prime. And deoxyribose is the sugar used for DNA, but for RNA, the sugar which is used by the molecule is different. It is basically ribose. And you can see that the difference primarily is at this point, where the oxygen group is missing in the case of the sugar in DNA, but not the sugar in RNA. Here is how the nucleotides are coming together, and this is done by basically by DNA polymerase. It is taking a nucleotide at a time and putting them in a very specific sequence, one after the other. And this is usually done when it's copying of the other strand, something I'll show you. And you can see the release of a proton and the release of a diphosphate group each time this is happening. I'll strongly urge all of you to do this homework. I would ask you to draw all 4 nucleotides, draw a tumor that is put 2 nucleotides together, and see how you lose two phosphates and a proton, and also draw a schematic of an antiparallel double helix. Here is a here is again a picture showing you the phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar backbone, and the nucleotides, in this case adenine, sticking out from the sugars, which makes up one strand of the DNA double helix. This is something you can practice. It's useful for you to do so.
So here I'm going to demonstrate drawing the simple structure of DNA. And now you don't need to be an artist to do this. So what the IB cares about is that you get the relative positions of the phosphate group, the deoxyribose sugar, both of those known as the sugar phosphate backbone, and the nitrogenous bases. Relatively, they need to be in the correct positions. Now, as I draw the second strand here, please note that it is anti-parallel. My deoxyribose pentose sugar is effectively like pointing down. This is somewhat difficult to draw on a computer screen. For you, what you might want to do is actually turn the page upside down, so you're drawing the same shape, it's just in the opposite direction. Now, once you have drawn it, you want to make sure that you label everything. So that's the hydrogen bonds, the covalent bonds, the nitrogenous bases, deoxyribose, the phosphate group, and go ahead and draw a line around a nucleotide as well.
So this gives you an idea of the biochemistry as well as the structure of DNA. And this is something you should sit down and draw. So let me move on now in the last 5 minutes to the last topic, which is basically the variety of DNA forms. Now, so Rosalind Franklin could move between these two forms, which gave different patterns on fiber diffraction images by just changing the humidity of the chamber in which the DNA was there. So in other ways, so to emphasize, if this particular strand of DNA which you are seeing over here, this B form of DNA, had a sequence, and I'm just going to write down a random sequence. This doesn't have any meaning. A, T, G, C, 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 A. If this was the sequence, the same sequence could make B form of DNA or A form of DNA, depending on the humidity of the chamber and the salt concentration which was there in the solution in which the fiber was sitting. Now, the 3 major forms of DNA, there are many forms of DNA which have been discovered over the years, but the classical forms are basically B form DNA, A form DNA, and Z form DNA. B and A form basically are right-handed, whereas the Z form is kind of left-handed. And the handedness can be seen if you look down the axis and follow 5 prime to 3 prime, whether it is turning, it's a right-handed, it's going in a clockwise fashion or in an anti-clockwise fashion. There is something called as a major groove, and this is an architectural feature which is over here. And there is something called as a minor groove, which is basically somewhere over here. Okay. The base pairs per helical turn, so this is a helical turn, are slightly different. As you can see, the A form of DNA is basically a little bit more compact. So this is one turn. And again, this is one turn. So you can see that the base pairs per turn seem to change. Okay. And there are other small differences which finally lead to a kind of different, let's use the term conformations of DNA, which you see. And there are biological implications of these different forms of DNA, which I will really not be talking about at this point.
Sir, please re-explain the groove part. What is exactly the groove?
Groove is that it is basically a large, let's call this a hole over here, uh, or it is a sort of a docking site. So this would be the major groove of DNA, this one, architecturally. Okay. And in fact, if you see over here, we know that there is a triple helical form of DNA which also exists. And if you look carefully at the triple helical form, specifically if you look at the way the yellow is, the third helix is coming, you'll realize that it actually sits in this major groove like this. Okay. So the major groove is an architectural feature, and it is actually used by proteins which are docking to DNA. The B form of DNA has a very large major groove. Okay, so over here this is the major groove, and this is the minor groove.
And sir, and what is exactly meant by anti and syn glycosyl bond conformation?
Okay, so this is basically basic chemistry. Can you just do a search, Google search, as to what anti and syn means?
No,
It's
no, sir, I understand the meaning, but here what
it's
does
the
it
same
imply?
meaning. There is no difference in the meaning. Remember, DNA is nothing but a chemical entity. So the
Okay, okay,
same definition for anti and syn, which is there in chemistry, is the definition over here.
okay, okay, sir.
Okay?
So there are 10 base pairs per helical turn in B form, right? There
Yes.
are written 10.5.
Okay, so I have got this from a textbook, and I guess there are small variations between textbooks. Remember, DNA is a floppy, flexible entity, which is a dynamic entity. It is constantly jumping around. Okay, so
Okay, sir.
measurements of different people may change slightly. Okay, let me end by talking about size scales. I just want you to appreciate the volumes of these molecular entities. On the right hand side, I have explained very basic things which you already know, that a nanometer is 10 raised to 9 meter, and a micrometer is basically 10 raised to 3 nanometers. The unit angstrom is used in crystallography quite a lot. 10 angstrom is basically 1 nanometer. All right. Now, this is the B form of DNA, 60 base pairs. You know what a base pair is now. And it will basically be about 204 angstroms in length. These are three proteins: a small protein lysozyme, a middling protein DNA gyrase. You can see all these holes in DNA gyrase where DNA will actually slide through these holes. And there is a large protein over here, which is one of the largest known in the cell, which is the ribosome. So you can appreciate volume scales and size scales by looking at DNA with respect to gyrase, lysozyme, and ribosome. Now, DNA is not naked in the cell, especially in the eukaryotic cell. It is wound around a set of proteins called nucleosomes. And on the left hand side, you see a picture of these nucleosomes and 2.5 turns, which is what DNA wraps around octamer of the nucleosomes. These octamers can slide along the DNA, which is also a very interesting feature of DNA. And at the bottom is a more realistic representation rather than just a ball and stick representation of the histone octamer and the linker DNA between them. The histone H1 acts as a sort of a clip making sure that the octamer does not fall apart. And this naked DNA is then folded around the nucleosome. Nucleosomes themselves fold into higher order chromatin structures. And these chromatin fibers keep on folding into higher and higher order structures, finally making up a chromosome. So I'll stop over here